Welcome to Module 2 of our course on Metabolism of Lipids. In Module 1, we covered lipid definitions, classifications, digestion, absorption, storage, oxidation, and key metabolic disorders. Here in Module 2, we'll focus on lipid biosynthesis and integration, exploring the pathways that build lipids from precursors, the hormonal and nutritional control of these pathways, and their connection to overall metabolic homeostasis. We'll examine fatty acid synthesis, ketone body formation and degradation, synthesis of complex lipids, triacylglycerols, glycerophospholipids, and sphingolipids, cholesterol synthesis and metabolism, steroid hormone biosynthesis, integration of lipid metabolism with other macronutrient pathways, especially the central role of acetic acid, acid-CoA. 1. Fatty acid synthesis. While beta-oxidation breaks down fatty acids to generate energy, fatty acid synthesis builds them up for storage and structural use. This process occurs primarily in the cytosol of liver cells, adipose tissue, and lactating mammary glands. 1.1. Precursor, acid-CoA. The carbon source for fatty acids is acid-CoA, produced in the mitochondria from carbohydrate and fatty acid catabolism. Because the mitochondrial inner membrane is impermeable to acid-CoA, it is shuttled into the cytosol as citrate, which is then cleaved back into acid-CoA by ATP citrate lyase. 1.2. The committed step. Acid-CoA is carboxylate to malonyl-CoA by acid-CoA carboxylase, ACC, the rate-limiting enzyme of fatty acid synthesis. This enzyme is regulated, activated by citrate and insulin, inhibited by palmitoyl-CoA, feedback inhibition, and AMP-activated protein kinase. 1.3. Fatty acid synthase complex. The multi-enzyme fatty acid synthase catalyzes a cyclic series of reactions. Condensation of acid and malonyl groups. Reduction using NADPH. Dehydration to form a trans double bond. Reduction to a saturated chain. Each cycle extends the fatty acid chain by 2 carbons, typically producing palmitic acid, C16:0, as the final product. 1.4. Elongation and desaturation. Palmitate can be elongated in the endoplasmic reticulum and mitochondria. Desaturation, introducing double bonds, is catalyzed by fatty acyl-CoA desaturases, but humans cannot introduce double bonds beyond carbon 9, hence the essential fatty acids linoleic and linolenic acids must come from the diet. 2. Formation and degradation of ketone bodies. Ketone bodies, acetoacetate, beta-hydroxybutyrate, and acetone, are water-soluble fuels derived from acid-CoA, produced mainly in the liver during fasting, starvation, uncontrolled diabetes, or high-fat/low carbohydrate diets. 2.1. Ketogenesis. In the liver mitochondria, acid-CoA condenses to form acetoacetyl-CoA. This is converted to HMG-CoA. HMG-CoA lyase cleaves it to acetoacetate, which can be reduced to beta-hydroxybutyrate or spontaneously decarboxylated to acetone. 2.2. Ketolysis. Extrahepatic tissues convert acetoacetate and beta-hydroxybutyrate back to acid-CoA for energy production. The liver lacks the enzyme succinyl-CoA acetoacetate-CoA transferase, so it cannot use ketone bodies itself. 2.3. Clinical relevance. Excessive ketone body production leads to ketoacidosis, a dangerous drop in blood pH seen in untreated type 1 diabetes and prolonged fasting. 3. Synthesis of triacylglycerols. Triacylglycerol, TAG, synthesis occurs mainly in the liver and adipose tissue. 3.1. Pathway. Glycerol 3-phosphate is formed from dihydroxyacetone phosphate, DHAP, in glycolysis, or from glycerol via glycerol kinase in the liver. Sequential acylation produces phosphatidic acid. Dephosphorylation yields diacylglycerol. Final acylation produces triacylglycerol. 3.2. Storage and transport. In the liver, tags are packaged into VLDL particles for transport to adipose tissue. In adipocytes, they are stored in lipid droplets. 4. Synthesis of glycerophospholipids. Glycerophospholipids form the structural backbone of cell membranes and serve as precursors for signaling molecules. 4.1. Key examples. Phosphatidylcholine, lecithin. Phosphatidylethanolamine. Phosphatidylinosytol. cardiolipin, mitochondrial membrane. 4.2 Pathways. Synthesis involves activation of either the head group or the diacylglycerol via CDP derivatives. These reactions occur in the endoplasmic reticulum. 5. Synthesis of sphingolipids. Sphingolipids have a sphingosine backbone rather than glycerol. 5.1 Pathway. Serine and palmitoyl-CoA condense to form sphingonin. Acylation produces ceramide. Addition of head groups forms sphingomyelin or glycosphingolipids, cerebrocytes, gangliocytes. 5.2 Clinical relevance. Inherited defects in sphingolipid degradation lead to sphingolipidosis, such as Tay-Sachs and Gaucher's diseases. 6. Cholesterol synthesis. Cholesterol is both a structural component of membranes and a precursor for bile acids, steroid hormones, and vitamin D. 6.1 Pathway overview. Acetyl-CoA condenses to form HMG-CoA. HMG-CoA reductase converts HMG-CoA to mevalinate, the rate-limiting step. Mevalinate is converted to isoprenoid units. Isoprenoids assemble into squalene. Squalene cyclizes to form cholesterol. 6.2 Regulation. Inhibited by cholesterol, feedback, and statin drugs. Activated by insulin. 7. Steroid hormone biosynthesis. Steroid hormones are derived from cholesterol and include glucocorticoids, e.g., cortisol, mineralocorticoids, e.g., aldosterone, sex hormones, e.g., testosterone, estrogen, progesterone. The pathway involves cleavage of the cholesterol side chain by desmolase, followed by specific hydroxylations. 8. Integration of lipid metabolism. Lipid metabolism is interconnected with carbohydrate and protein metabolism. 8.1 Central role of acetyl-CoA. Acetyl-CoA is the key precursor for fatty acid, cholesterol, and ketone body synthesis, while also being a product of carbohydrate breakdown via glycolysis and pyruvate dehydrogenase. 8.2 Fed state. High insulin promotes fatty acid synthesis and tag storage. Glucose is converted to fatty acids via acetyl-CoA. 8.3 Fasting state. Glucagon and epinephrine stimulate lipolysis. Fatty acids are oxidized to acetyl-CoA, which fuels the TCA cycle or is diverted to ketogenesis. We have now covered the complete picture of lipid metabolism. Module 1, Lipid Basics, Digestion, Absorption, Oxidation, Disorders. Module 2, Lipid Biosynthesis, Ketogenesis, Synthesis of complex lipids, cholesterol and steroid hormones, and integrated metabolic control. Understanding lipid metabolism is crucial not only for biochemistry but also for medicine, nutrition, and public health, because disturbances in these pathways underlie many common diseases from obesity to cardiovascular disorders.